So, in today's lecture, we will continue to discuss the concept of receptors as drug targets. In the last lecture, we discussed about how agonists work and how one can understand the various aspects of how agonists can activate a receptor. So, today we will look at the other concept of antagonists. So, antagonists are as the name suggests are molecules which can sort of completely or abrogate the activity of a receptor. So, the way we understand antagonists is that, so let us say you have the natural substrate that is binding to the receptor and activating the signaling cascade. Now, if you have an antagonist drug, what it does is it goes and binds to the same receptor site, and in the process it blocks it. So, therefore, it is a way in which you can prevent the natural messenger from binding to the receptor. So, just to recall, the way in which we understand receptor binding is through what is known as an induced fit. So, the messenger binds and then the receptor undergoes a conformational change, along with it the messenger also undergoes a conformational change, and once it binds, then the signal is sent forward, and after that the messenger dissociates and the receptor goes back to its original shape. So, now let us look at the principles of an antagonist, how to design an antagonist, and we looked at in the previous lecture how to design drugs that can activate the natural receptor or activate a receptor to obtain a biological effect. Now, what we would need to do in this case is that we would need to figure out how to address a problem where there are too many messenger molecules acting on the particular receptor. So, what happens in this process is that you would have hyperactivation or large scale activation of the receptor, which leads to unwanted excess signaling. So, we need to design a drug that can counteract this process. So, the answer would be to design an antagonist that will bind to the binding site, but will not activate the receptor. Okay. Now, since it is bound, it will prevent the normal ligand from binding and activating the receptor. So, keep in mind we have looked at how enzyme inhibitors can be designed. So, for example, a competitive inhibitor will go and bind to the enzyme, but there is no turnover. Okay. So, here in this case an antagonist will similarly bind to the receptor, but there would be no signal that is transmitted. Okay. So, therefore, one has to keep in mind this concept while designing an antagonist. So, there are several strategies which we can employ to design an antagonist, but one of the ways is to design a drug that is in the right shape to the receptor binding site, but will either not induce the conformational change or it will distort it in the wrong way. Okay. So, let us look at this example here. So, recall our hypothetical messenger. So, it had a hydrogen bonding over here and a van der Waals interaction over here and an ionic interaction over here. So, all these three interactions are necessary for the chemical messenger to induce a conformational change to give you the signal. Okay. Now, in the case of the antagonist, what one could do is, if we could elongate this chain a little bit. So, keep in mind that this is where the binding happens, but now if you could elongate this change a little bit, this conformational change that we would expect from here to here may not happen. Okay. If that conformational change does not happen, then downstream the effects do not occur. So, this is one way in which you could design an antagonist. Okay. So, to recap, what we would need to do is to design a molecule that does not induce the conformational change, but yet binds to the receptor site. Okay. One more strategy is to be able to use binding regions which are not used by the natural chemical messenger. Okay. So, another strategy is to find different binding regions within the binding site that are not used by the chemical messenger. So, recall in our previous hypothetical receptor, there was a hydrophobic binding region, there was an ionic binding region and a hydrogen bonding region. Now, all these three are going to bind to the neurotransmitter. However, if you look at the biochemical structure of the receptor, you will find that there is an extra hydrophobic region that can potentially be used. Okay. Now, when the extra binding regions are involved, it may induce a different fit. Okay. And once this different fit is induced, it is possible that that may not be giving out the same conformational change that was necessary for the signal to be generated. Okay. So, extra binding regions do not necessarily have to be within the binding site. It could be something that is quite a little bit away from the binding site, and that can be exploited to design an antagonist. It is also quite common to find antagonists that are larger than the natural messenger. So, if they are larger, what happens is that they can access extra binding sites beyond the reach of the usual messengers. Okay. And many antagonists are capable of binding to both the normal binding site and these neighboring regions that are around this receptor binding site. So, let us look at this example that we sort of started out with. So, here is the natural ligand, and what happens is that once it binds here to the hydrophobic region, and then there is a hydrogen bonding effect that is here, and this is the ionic bond. Okay. Now, what happens in this process is that this, in order to achieve better binding, the pocket moves in and undergoes, and in doing so it undergoes a conformational change, and this conformational change results in a signal being transmitted. Now, if you were to design a molecule that exploited this extra hydrophobic region here, what happens is that the fit of the molecule is such that it binds to the hydrophobic region, it still involves in hydrogen bonding, but the only difference is that because it is already bound to the hydrophobic region, this interaction is still happening, but does not require the protein to undergo a conformational change. So, the three important binding regions are still present, but the extra hydrophobic region which acts as a potential binding region does not require a conformational change or does not induce a conformational change. Okay. So, what happens is that if we have designed a more potent agonist, but in this case, since the induced fit is significantly different, the receptor is not activated. So, this is one principle that one could employ to design a new antagonist. Now, because the antagonist is now bound to the active site or to the binding site of the receptor, it prevents a new molecule or new chemical messenger from coming and binding to it. And so, because it prevents the new one from binding, it acts as a inhibitor or an antagonist. Okay. So, antagonists that bind strongly to the target binding site are often used to label receptors. Okay. So, the way we understand the receptor, the topology and the structure inside a receptor is we have to crystallize this molecule in the presence of either the natural ligand or an antagonist. So, once we are able to crystallize it, then we know what are the kind of interactions that happen inside, and we may be able to understand the landscape in the binding site. Such antagonists are synthesized typically with a radioactive isotope incorporated into the structure. So, what this allows us to do is to be able to detect this the presence of this antagonist. Okay. So, the hypothesis here, the working hypothesis here is that we should be able to design drugs if we have intimate knowledge of the binding site, which is not easy to attain, and it is not as straightforward as it sounds. For many years, the only way to map out the receptor structure was to synthesize and study various compounds that interacted with this receptor. So, we would change the benzene ring to a cyclohexane, or put in a substituent and the ortho, meta or para position, or change the alcohol to an amine or a carboxylic acid, and so on and so forth. So, by systematically synthesizing these molecules and studying how they interact with the receptor, then what we would be trying to do is to solve a 3D jigsaw puzzle, and we would try and find out where each sort of, or what are the various binding site, potential binding sites inside the structure of the receptor. In the past several decades, computational studies have significantly helped, because once we have some sort of an X-ray structure, then we would be able to predict what kind of binding sites that are present in the receptor and so on. So, these concepts we will look at later in the course. As with enzymes, you also have a possibility of allosteric modulation. Okay. some receptors have allosteric binding sites, and these are binding sites which are located on a different part of the receptor surface, and they bind to the natural molecules called as modulators. And these modulators modulate the activity of the receptor by either enhancing or diminishing it. So, again one can draw a parallel to what we have learnt in enzyme catalysis, in that we also have allosteric binding sites which can help regulate the activity of an enzyme. Now, if the activity of the receptor is diminished, then the modulator is indirectly acting as an antagonist. So, in order to understand this concept better, let us look at a cartoon. So, here is an example of an allosteric modulator, and here is the binding site for the receptor, and once this allosteric modulator binds, this receptor is shut. Okay. So, again similar to how an enzyme allosteric modulation of an enzyme occurs, the main binding site of the receptor is not in play here. It does not directly bind to the modulator, but once the modulator binds to a site which is remote from the binding site, this induces a conformational change at the binding site which shuts it. Okay. So, if the site becomes too distorted, then it is no longer able to bind the normal chemical messenger, and therefore, there is no activity, right. So, this is how we understand allosteric modulators. There is also another effect which is called as the umbrella effect. Okay. As the name suggests, an umbrella can provide shade to an individual, but if it is large enough, then it can also provide shade to multiple individuals. And so, similarly, when an antagonist binds to this allosteric site, let us say, now what happens is that it can act as an umbrella and not just interact with this neighboring site, it also blocks the binding site. Okay. So, although they do not bind directly to the binding site, this molecule acts as a shield or as an umbrella, preventing the normal messenger from acting on the binding site. So, this effect is called as the umbrella effect. There are frequently when in drug discovery, we come across what are molecules which are known as partial agonists. Okay. So, these are molecules which cannot be defined either as a pure agonist or a pure antagonist. So, the compound acts as an agonist and produces a biological effect, but that effect is significantly smaller than what we would get with the normal ligand. Okay. So, therefore, it is called as a partial agonist, right. Now, let us look at this example over here. So, again recall the natural the hypothetical messenger that we used, and we have here the same or a very similar interactions that are happening. So, here is the hydrophobic interaction, here is the hydrogen bonding, and here is the ionic effect. Okay. So, once this molecule binds to the receptor site, this channel which was blocked is now open, but it is not completely open. Okay. So, it results in some partial opening. So, imagine that this is an ion channel receptor. Okay. So, once it binds to the receptor, it can open it, but which will permit the entry of ions, but it is not completely open. So, therefore, it is a restricted entry into the cell. So, what happens is that the rate at which the signal is transmitted could go down. So, this is known as a partial agonist. Okay. So, partial agonists may be capable of binding to a receptor in two different ways by using different binding regions in the binding site. Okay. One method of binding activates the receptor, that is called an agonist effect, but the other does not, which is called the antagonist effect. So, the balance of agonism versus antagonism would then depend on the relative proportions of molecules binding by either method. Okay. So, we will look at later in the lecture how we can understand this by some certain plots. Receptors that bind the same chemical messenger are not all the same. The partial agonist may be capable of distinguish between different receptor types or subtypes. So, for example, we looked at earlier how a particular receptor in the heart may be structurally different from a particular receptor in the lung, and that receptor, because of its subtle differences, can bind to a partial agonist in a way that is very different from the other receptor. So, in doing so, what may happen is that the partial agonist may work better on one receptor subtype compared to the other. That is, it can act as an antagonist for a particular receptor, or it can act as a agonist for a particular receptor, while it can have a different activity on the same receptor subtype. Okay. Now, let us look at how to understand these drug receptor interactions better. Okay. So, in order for us to appreciate the mechanisms of drug action, it is important for us to understand the forces of interaction that bind these drugs to their receptors. Again, we have looked at in detail the various forms of non-covalent interactions, but just to recap, you can have hydrogen bonding, you can have dipole-dipole interactions, you can have ionic interactions, you could have hydrophobic interactions, you can have van der Waals interactions, and so on and so forth. Of course, there are covalent interactions which we will not come across very frequently in drug receptor interactions, but these non-covalent interactions will play a big role. Okay. Now, because of the low concentrations of drugs and receptors in the bloodstream, the law of mass action alone cannot account for the ability of how small doses of these specific drugs elicit such a large difference in the biological system. Okay. So, what we need to understand is how this amplification effect happens. So, in order to understand that, let us define certain terms now. So, the stability is of the is usually defined by an equilibrium constant, which we shall call as Kd, okay, which is known as the dissociation constant of the drug receptor complex. The dissociation constant is defined as the concentration of the drug here multiplied by the concentration of the receptor divided by the drug receptor complex. Okay. So, very simply, the larger the dissociation constant, the drug receptor complex is going to be in a greater concentration. Now, which also means that the drug has a very high affinity for the receptor. Okay. So, typically a large number of studies show that the median dissociation constant for inhibitor drugs in the market is around 20 nanomolar. Okay. So, we have molar, divide that by 1000, you get millimolar, and then again divide that by another 1000, you get micromolar, and then divide that by another 1000, you get nanomolar. So, nanomolar is extremely small concentrations inside the body. Okay. Now, let us this for the inhibitor to act at this low concentration, we need to understand the factors that determine the drug receptor affinity. Okay. So, as we have studied previously, the free energy is related to the equilibrium constant in the following way: delta G naught equals minus RT ln KEQ. So, the spontaneous formation of a bond, that is here a non-covalent bond, results in the decrease in the free energy and will occur only when there is a negative free energy of interaction. Okay. So, and this negative delta G is related to the enthalpic term as well as the entropic term. So, that is we are already familiar with, delta G equals delta H minus T delta S. So, with this background, now let us look at inverse agonists. Okay. So, many antagonists that bind to a receptor binding site are in fact more properly defined as inverse agonists. Okay. So, this inverse agonist has the same effect as an antagonist, but it fails to activate it. Okay. So, what this means is that there is some residual activity that is there in the receptor to begin with, and this is going to be modulated by the inverse agonist. So, this was a very important finding, because what we have been able to understand from this is that the receptors have constitutional activity and an inverse agonist is capable of preventing this activity. So, there are some receptors such as GABA, which is gamma aminobutyric acid receptor, serotonin, dihydropyridine receptors, which have a low level of inherent activity even in the absence of chemical messenger. So, that means that these are constitutional activities that is already going on inside the cell. Okay. Now, what an inverse agonist does is that once it binds, it prevents the activity from happening. Okay. So, again we will look at this later in the course. Okay. So, the discovery that some receptors have an inherent activity has some important implications in receptor theory. it suggests that these receptors do not have a fixed inactive conformation, but are continually changing shapes such that there is an equilibrium between the active conformation and the different inactive conformations. Now, in order to understand this, let us look at some cartoons. Okay. So, for in a traditional receptor, there is a resting state which is shown by these gray pictures here. Now, what happens is that it is in equilibrium with the active conformation, which is what our inverse agonist tells us. And once you add an agonist, what happens is that you have the active conformation in a larger concentration. So, once the active conformation is in a larger concentration, it results in enhanced signal transmission. Similarly, when you add an antagonist, so this antagonist that is added here goes and binds to the both the active as well as the inactive conformation, and once it binds to the active conformation, it pushes the equilibrium in the direction of the inactive conformation, and therefore, the signal goes down. Okay. An inverse agonist is proposed to have a binding preference for the inactive conformation, and once it stabilizes the inactive conformation, it shifts the equilibrium away from the active conformation. Okay. And so, therefore, there is a drop in the biological activity. Now, we looked at the concept of a partial agonist, and in order to understand this, what we can again propose an equilibrium between the active and the inactive conformations, and the partial agonist binds to the active conformations as well as the inactive conformation, and what it does is that it allows for this equilibrium to occur, wherein only a diminished signal is manifested.